Welcome back to our talk about cellular respiration. So we are going to complete all your information about cellular respiration today. So this is where we left off. So far we have completed glycolysis within the cytosol of the cell, and that has created pyruvate, which we then put through the preparation reaction, which happened in the mitochondrial matrix. And so far we have produced acetyl-CoA, which we know is going to be fed into the citric acid cycle. So that's where we stand. That's where we left off. So one thing I figure you might be wondering is we have been talking about all of this energy production by starting with glucose. And I know that you are thinking, I don't eat straight sugar, which of course could be sucrose or fructose, and so where does all this other food that I eat play into this? So I wanted to take a moment just to stop right here to talk a little bit about this, and of course it's not going to encompass that entire question, but as far as acetyl-CoA goes, it can come from a various amount of sources. First is the glucose catabolism through glycolysis that we've been talking about, and so that is our straightforward answer. The second one is from fatty acids through a fatty acid oxidation cycle, and a third one is glycogen, which can be catabolized and then sent through glycolysis. Okay, so we, of course, are going to eat fats. Our body, when we have extra energy to store, is going to store it either as glycogen or fat. And so glycogen is a much more rapid digestion. And so if we fast, our body can use its glycogen stores and have enough energy for about a day. Our fatty acids, which are normally stored in adipose tissue or fat tissue, those storages can, they store more energy, and we can live for several weeks, maybe even a month on those fat stores. So the difference is the fatty acids are a little harder to break down. But this image right here on the inset, you'll see some lipid droplets. So our fatty acids are stored in here. This one happens to be in cardiac tissue. Cardiac tissue does tend to store fatty acids. And I don't need you to know the details of the fatty acid oxidation cycle, but at this point you know what oxidation is. And so the breakdown of fatty acids go through a whole series of reactions to come up with our acetyl-CoA. So this is one source of acetyl-CoA that's going to go into the citric acid cycle. So the other form of storing energy is glycogen, and this is for animal cells. And so glycogen is stored in the cytosol of various cells. Particularly in humans, the liver cells store a lot of glycogen, and it's highly regulated. Its catabolism and anabolism are severely regulated in the liver. And so when that is called upon, because the body needs some more energy, it's much faster to break down glycogen than it is to break down fat. So usually those resources are pulled first. So glycogen is broken down into glucose 1-phosphate, which is a 6-carbon sugar, and then that's fed through glycolysis. So just like glucose is broken down through glycolysis, glucose 1-phosphate is also broken down through glycolysis. And through that, 2 pyruvate molecules are produced, and those go through the typical preparatory reaction to produce 2 acetyl-CoA. So what you may have noticed is our glycogen can go through glycolysis, and so we can take glycogen and break it down into sugars and then into acetyl-CoA. Our fatty acids cannot be broken down into sugars, of course, so it has to go through a different process in order to produce acetyl-CoA. And then from there on, the rest of it can be metabolized through the citric acid cycle. So that is how we get to acetyl-CoA, and now we can proceed through the rest of cellular respiration. So for the citric acid cycle, what we're going to start with is here on the left. For every glucose molecule, we have 2 acetyl groups, 6 NAD pluses, 2 FAD, 2 ADP, and 2 inorganic phosphates. And they're going to go through the citric acid cycle, and what we're going to come out with on the end is 4 carbon dioxide molecules, 6 NADHs, 2 FADH2s, and 2 ATPs. So this is the overall view of what's going in and what's coming out. And the citric acid cycle is exactly that. It is a cyclical metabolic pathway. And so at the end, we're going to recycle out some of our inputs, but what other products are coming out are going to be moving along to the electron transport chain. So the citric acid cycle is going to take place in the matrix of the mitochondria. So just to back up a little bit, we started with our glucose molecule, 6 carbons, and it's gone through glycolysis to produce pyruvate, and then through the preparatory cycle so that pyruvate is then processed through and we come up with acetyl-CoA. So that's our starting point right here for our citric acid cycle. So acetyl-CoA donates that acetyl group to a 4 carbon molecule called oxaloacetate. So that's right here. And those two combine to make citrate, or citric acid, which is where the citric acid got its name from, citric acid cycle. Okay, now this whole metabolic pathway goes through a series of 7 different enzyme complexes, and we're basically going to oxidize all the carbon and have the opposite reduction reactions occurring as well. So remember that we came in originally with 6 glucose molecules, and in our preparatory reaction we gave off 2 carbon dioxide molecules. And yes, I know I do not write very well with this pen. Hopefully you can read that. Okay, now with 1 rotation of the citric acid cycle, we're giving off 1, 2 carbon dioxide molecules. Okay, well, keeping in mind then that for every glucose molecule we go through the citric acid cycle twice, that actually means that we've produced 4 carbon dioxide molecules. Ooh, does that look like a 4? Yikes. Okay, so this is where the carbons go. Having started with 6 carbons in glucose, we've now oxidized them and broken them down into 6 carbon dioxide molecules, which can then diffuse across the cell membrane, out into the blood, out to the lungs, and then we exhale that carbon dioxide. So that's the oxidation side of the reaction. What's the reduction side? Okay, for the reduction side, we have our carriers that we talked about that were so important. We have NAD plus that gets reduced to NADH. And NADH is a high electron carrier. It's one of those intermediates, and that's going to carry those electrons to the electron transport chain. And we're going to talk about that in a little bit. But let's look at our citric acid cycle. We've produced 1, 2, 3 NADHs for 1 time around the cycle. So, like I said, for every glucose molecule we go around twice, so we've produced 6 NADH. And I don't even know if I can type all that with this silly pen. Ugh, that's as good as it gets. Okay, the other intermediate carrier that we talked about was FAD. So we've also produced FADH2. a high electron carrier. And for 2 times around the cycle, we have 2 FADH2s produced. Y'all get the point here. Sorry, I'm really slow at this part. Okay, so far we've produced a total of 6 carbon dioxide molecules, but most importantly, we've produced 6 NADH molecules and 2 FADH2s that we're going to use in the next section. Something to keep in mind is that we have regenerated oxaloacetate so that the cycle can continue to go on as glycolysis moves forward, more glucose coming in. The other thing that you'll notice is that we've produced some ATP. In fact, we produce 2 ATP molecules for every glucose molecule involving the citric acid cycle, and that is through substrate level phosphorylation that we talked about before. So what was the point? The point of the citric acid cycle is to use acetyl-CoA to make reduced high-energy intermediates. And those high-energy intermediates are our NADH and our FADH2. And so there are other byproducts such as carbon dioxide gas that we talked about and small amounts of ATP. So it seems like we've been through a long process and still not made a lot of ATP. Well, that's where the next process comes in, and it makes it all worthwhile. So the electron transport chain, this is where the action gets good. This is where we start producing lots of energy. So let's look into that really closely so you understand it. First of all, where does the electron transport chain even take place? So all of this is located in our mitochondria in the inner membrane. All those cristae that make so much surface area allow for lots and lots of ATP to be made. So we're going to be looking at this inner membrane of the mitochondria. So who are the players in the electron transport chain? The first are the high energy electron carriers that we've talked about ad nauseum probably, NADH and FADH2, and those are coming from the citric acid cycle with high energy electrons. The second are the redox carriers. Those are the proteins that are embedded in the intermitochondrial membrane. ATP synthase, which we've seen before in our photosynthesis reaction, and oxygen is our final player. Many of our redox carriers, or our proteins that undergo electron transport, as in passing them from one carrier to the next, are what we call cytochrome proteins. And cytochrome proteins have at the very core an iron atom. And that iron, when it accepts electrons, it becomes reduced. If it gives up its electrons, it becomes oxidized. And as we'll see, it cycles through this oxidation reduction, oxidation reduction, oxidation reduction, as electrons are passed down the electron transport chain. Okay, so let's talk about how this works. First, let's get our bearings straight. So we're in the mitochondria. Here is the inner matrix, right? And then here is the inner membrane. And so this is that small space between the inner membrane and the outer membrane. So lots of surface area on this membrane, and so this would be occurring in multiple places, but we're just going to look at kind of one section at a time. So we've talked about our NADH and our FADH2. Those are our electron carriers. And so they're bringing in these high energy electrons. And so you'll notice all in the membrane are these proteins, reductases and oxidases. And so remember that we are passing off these electrons. We're reducing and then oxidizing, reducing and then oxidizing. various proteins. In this particular case, it tends to be a cytochrome reductase or oxidase. So to begin with, our NADH is going to give up this high energy electron to what we call NADHQ reductase. So that's this protein right here that spans the membrane. And notice that FADH2 is also giving up its electrons to an enzyme called coenzyme Q. And so as we give up our electrons, the NADH and the FADH are being oxidized because they are giving up electrons. So these two proteins in the membranes then are being reduced. They're gaining electrons. They're gaining that negative charge. They're being reduced. Okay, then we move on down the membrane, and here is another, so here is another enzyme that is then gaining this electron, so it's being reduced. And all throughout here, notice that when we give up an electron, a hydrogen ion is then being passed through that protein because that energy is being used to pump hydrogen ions against the concentration gradient. So we're building a concentration of hydrogen ions outside of this membrane. And so we continue to do that, passing it down. Here's another cytochrome C, another cytochrome oxidase. So every time we're giving up electrons, and as we pass it down, that high energy electron is starting to lose its energy, right? Because it's being used over and over again to pump hydrogens across the concentration gradient. Now keep in mind, of course, this isn't one single electron, right? We're doing this multiple times. But we're reducing that energy because we're using it to pump hydrogen across the a concentration gradient, so pumping it. And when we get towards the end, we need a final electron acceptor. And what we find in the mitochondria is oxygen is the primary electron acceptor. And so when we then give oxygen this electron, it's the final reduction, right? Then it joins with some hydrogen ions to make water. So that's our final product. Now, what we've accomplished here is lots of hydrogen ions here and not so many here. So that concentration gradient itself is a huge form of potential energy. And that potential energy is used by the ATP synthase down here. And so as the hydrogen ions are released back into the matrix, ADP plus a inorganic phosphate is then put together using the energy that was built up from this concentration gradient to then make ATP. And this happens over and over and over again. So that is how we make ATP within the mitochondria. Okay, so this is a close-up look of our ATP synthase. This is our enzyme that is the ultimate producer of ATP within a cell. And notice that it is an integral protein. We've talked about that. And here is our hydrogen ion concentration high within the inner membrane space. And so the different parts of ATP synthase means that there's a rotor within the membrane, and that's this part here. And it spins clockwise when the hydrogen flows past it down the gradient. So as the hydrogen comes through, it spins this rotor. Now, there's a stator, or is what it's called, that anchors the whole complex into the membrane, and that's right here. Then there is the rod, or the stalk, and that extends into the knob, but it also spins and activates the catalytic sites in the knob. This right here is the knob. And there are 3 catalytic sites, or 3 active sites in this particular enzyme, and all 3 of them join inorganic phosphate to ADP to make ATP. So as the hydrogen ions come through, and spins the rotor, we produce 3 ATP molecules. So that's what our ultimate goal is producing lots of ATP. And that comes from basically this motor that is driven by a hydrogen ion concentration gradient within the inner membrane of the mitochondria. I'd like you to take a minute and try to fill out what each of these letters represents on the electron transport chain. You will see this diagram again, so I'll go ahead and tell you that. The answers are on the next slide, so you can come back and test yourself anytime. Okay, so let's summarize it. So we have brought in, if we're looking at this, by the way, let me get you oriented to where we are. We have our mitochondria. We have done a cross section of it. So this would be the outer membrane here, the intermembrane space here, and the matrix here. And so this is our inner membrane of the mitochondria. So we have brought in pyruvate from glycolysis and fatty acids, and from those two we've gotten acetyl-CoA through our preparatory reactions, and then those entered the citric acid cycle, and we have our byproduct of carbon dioxide, and then we also have our high electron carriers, our NADH and our FADH2. And those are going to donate our electrons to a series of electron carriers that are contained in this inner membrane here. And while we do so, then we create a hydrogen ion gradient as hydrogen ions are pumped into that inner membrane space. And then our final electron acceptor is our oxygen, and so oxygen gets reduced to water. And then this concentration gradient can be used by our ATP synthase complex as hydrogen ions are leaked back through into the matrix, and that is combined to make ATP. And so then ATP can be transported out of the cell. So that is our summary of how the citric acid cycle and the electron transport chain all put together make a lot of ATP. So I've taken this image from your textbook so that we can look at the ATP production that we've talked about through cellular respiration and its different steps. So remember, we begin always with one glucose molecule, and so in the cytoplasm through glycolysis we produced 2 net ATP molecules. In the citric acid cycle within the matrix of the mitochondria we produced another 2 ATP molecules. So that brings our total to 4 ATP molecules so far. Now, these ATP molecules have been produced by substrate level phosphorylation. Then we move to the electron transport chain and we produce a lot of ATP, about 32 to 34 molecules of ATP. And this is through oxidative phosphorylation. So a slightly different process, more is produced. So those two together yield about 36 to 38 ATP molecules per glucose molecule digested. The cells in the human body that need to produce the most energy are those that you would expect, actually. Those in the brain, the heart, the liver, and the muscle tissue. In fact, liver cells can have up to 2,000 mitochondria per cell. So if you think about how much ATP can be produced there, that's pretty impressive. But the only cells in the human body that do not have mitochondria are red blood cells. And that's because at maturity, red blood cells basically get rid of all of their organelles. They are primarily there to transport oxygen, and so they don't need a lot of energy to do that. Everything we've talked about so far for our ATP production has been dependent on one particular molecule, and that is oxygen. So what we've just been through with cellular respiration is what we call an aerobic respiration process. Aerobic meaning oxygen is present and necessary. So we produced ATP through oxidative phosphorylation, with the last electron acceptor being oxygen. So what happens when oxygen is not present? We call this anaerobic respiration, or also what we call fermentation. So the fermentation process is the breakdown of glucose to make ATP under anaerobic conditions, and it can only undergo glycolysis. So let's talk about that for a minute. There are two types of fermentation. The first is lactic acid fermentation. So as this shows here, we're again starting with glucose. We go through glycolysis to produce 2 pyruvate molecules, and through that we produce 2 ATP molecules through substrate level phosphorylation. And in that we are going to reduce NAD to NADH. That NADH is going to be used to reduce pyruvate to 2 molecules of lactate, or what we call lactic acid. So there's no intermediates. Pyruvate accepts electrons straight from NADH. And so what we have here is a production of 2 ATP molecules. Now that falls way short of our oxidative phosphorylation from the electron transport chain, but the body and cells still need a way to produce ATP, even if it's small levels, just to be able to maintain life. So this is what fermentation looks like in the human body. You're very familiar with it. Normally, when you put a demand on your body, such as running, you begin to start breathing heavier because each cell has a higher need for oxygen because it needs to produce more energy so you can accomplish these goals. And so with cellular respiration, we need more oxygen for that last electron acceptor. So what happens when your muscle output needs to be more than the energy demand can be met? In other words, you need more energy at some point than you can produce because you can't bring in enough oxygen fast enough. Well, when we can't bring in enough oxygen, that's when fermentation kicks in, and particularly in these muscle cells that really require lots of energy production. When fermentation kicks in, we start building up lactic acid. And so you are familiar with that because that's what's going to make your muscles sore. It's because you have an energy demand that cannot be met. And so, interestingly enough, lactic acid, the way that we eventually get rid of that, it will build up in the muscle all throughout the fermentation process. Once you slow down and the demand lessens, and you continue to breathe in that oxygen that's necessary, then we can start producing ATP through our oxidative phosphorylation. And the lactic acid then is transported through the blood back to the liver, which is kind of interesting because the liver has more mitochondria than just about any other cell within the body. And so it uses that energy from the mitochondria then to break down the lactic acid. Even though lactic acid may not be pleasant for us and will hurt in our muscles, it's actually used in the food industry for flavor. So there are bacteria that go through anaerobic fermentation to produce lactic acid, and those are used to flavor our yogurts and our cheeses. So when you talk about probiotics in yogurt, you're talking about the bacteria that will go through fermentation to produce that sharp acidic taste that you taste in your yogurt. Second is alcohol fermentation, and this occurs often in yeast cells. Yeast cells do actually prefer aerobic respiration with cellular respiration, but when oxygen is not present, then they go through alcohol fermentation. So very similar, we still start with glucose and go through glycolysis to produce 2 molecules of pyruvate, and this is where we get our ATP production. So 2 molecules of ATP. And then in yeast fermentation, then our pyruvate is converted to this 2-acetaldehyde that is then reduced into 2 molecules of ethanol. So the byproducts of fermentation are going to be carbon dioxide and ethanol, or ethyl alcohol. So lucky for us, yeast cells in anaerobic conditions do go through fermentation, and we utilize the byproducts of that fermentation. And so we use that process when we bake bread. The carbon dioxide that's released allows for the dough to rise, and then with bread at least we cook off the alcohol. And then of course we capture that alcohol when we are making beer or wine. It's also used in making kombucha or in making vinegar. So lots of uses for this yeast fermentation, but the yeast, you actually use it to produce energy and to carry on life. So that is the end of our energy production unit. Hopefully all of that has made sense to you. Definitely review, make sure you know which enzymes are used when, what the reactants are, what the products are, what the intermediate electron carriers are, all those things. And then we'll meet back here next time.